Hello all, hope you're having a great day today. Today we are continuing our discussion in Chapter 7 over the water-soluble vitamins, and this is Part 4. Here we go. So folate or folic acid is a salt of folic acid form that is found in foods, is a water-soluble vitamin. It is part of the B complex. Folate is found naturally in some foods, but folic acid is a synthetic form found in fortified foods and dietary supplements. Collectively, they are called folacin. The RDA for a folate is given as micrograms of a dietary folate equivalents, or DFE. 1 DFE equals 1 microgram of food folate. 0.6 micrograms of folic acid in fortified foods, or 0.5 micrograms of folic acid supplements taken on an empty stomach. 1 microgram of folic acid in fortified foods equals 1.7 DFE, while 1 microgram of folic acid in a supplement equals 2 DFE. Because these forms are better absorbed, the RDA for folate is 400 micrograms of DFE per day. During pregnancy, the RDA is increased to 600 micrograms of DFE per day, and it is 500 microgram DFE during the early stages of lactation. The daily value is 400 micrograms of DFE, and an upper limit of 1,000 micrograms DFE per day has been set for adults, and even lower amounts for younger individuals. Folate derives its name from foliage, because it is found in green leafy vegetables such as spinach. Other good sources include organ meat such as liver and kidney, dry beans, whole grain products, and some fruits such as oranges and bananas. 1 cup of cooked black or navy beans contains about 60 to 75% of the folate RDA. 1 banana provides almost 10% of the RDA. Currently, all cereal and grain products in the United States are fortified with folic acid to reduce the occurrence of neural tube defects. The average fortification is 140 micrograms per 100 grams of food product. Studies indicate that such fortification has increased daily dietary folic acid intake by 100 micrograms or more. Folate serves as a part of a coenzyme that plays a critical role in the metabolism of methionine, an essential amino acid. In this regard, folic acid is critical to the formation of DNA, the genetic material that regulates cell division. It is essential for maintaining normal production of RBCs, one of the most rapidly dividing cells in the body. Folate is also critical during the very early stages of pregnancy when all cells in the fetus divide rapidly. Researchers indicate during periods of rapid cell division, large amounts of folate are needed to make DNA. Folate is also involved in the metabolism of homocysteine, an amino acid derived in the conversion from methionine and considered by some experts to be a possible risk factor for cardiovascular disease. Folate is necessary for the reformation of methionine from homocysteine. One national survey has reported folate intakes below the RDA for some Americans. Individuals who consume large quantities of alcohol and women who take oral contraceptives may experience deficiencies in folic acid, as these drugs may impair absorption of the vitamin A. Folic acid deficiency may impair DNA formation or lead to an increase in homocysteine. Due to its effects on DNA synthesis, one of the major effects of folate deficiency is pernicious anemia, attributed to an inadequate RBC regeneration. Anemia could impair delivery of oxygen and significantly impair performance in aerobic endurance events. DNA and chromosomal damage caused by folic acid deficiency has been suggested to increase the risk of cancer. For example, in a Tufts University review, a low level of folate in large intestine cells was associated with an increased risk of colon cancer, a finding one of the authors reported to be consistent with the results of 20 other epidemiological studies. which have found that people who eat the most folic acid are the least likely to get colon cancer. Zhang and others also reported that for women who consume alcohol, those with the lowest dietary folic acid intake were at the highest risk for developing breast cancer. Due to either DNA damage or the adverse neural effects of homocysteine, women who are folate deficient and become pregnant may give birth to children with neural tube defects, such as spina bifida, an incomplete closure of the tissue surrounding the spinal cord. Such defects may cause paralysis and severe disabling conditions in the child. Approximately 2,500 infants in the United States are affected each year. Elevated plasma levels of homocysteine are thought to damage the lining of the blood vessels or initiate growth of cells that form the framework of plaque, increasing the risk of several vascular diseases, including coronary heart disease, or CHD, stroke, peripheral vascular disease, and Alzheimer's disease. However, Wierzbicki contended that it is simply a marker for CHD, not a cause. As an aside, Jobert and Minor, in a review of exercise nutrition and homocysteine, noted that no consistent relationship exists between physical activity and plasma homocysteine concentrations. The American Heart Association has not classified a high blood level of homocysteine as a major risk factor for CHD. And you can see here the deficiency that there is an effect on exercise performance. As well, there could be, with anemia, it could impair endurance performance as well. In a review, Eichholzer and others reported irrefutable evidence that adequate folic acid helps prevent neural tube defects. In countries mandating fortification of flour with folic acid, the incidence of neural tube defects decreased by 31 to 78%. The NIH report on vitamin supplementation also noted that multiple studies have supported the effectiveness of folic acid use by women of childbearing age to prevent neural tube defects in offspring. All women who have the potential to become pregnant could consume 400 micrograms of synthetic folic acid daily. The 400 micrograms should be consumed as folic acid in fortified foods or dietary supplements, in addition to normal food folate intake. When pregnancy is confirmed, the RDA is increased to 600 micrograms DFE. Currently, only 1 in 3 women consume 400 micrograms of DFE daily, so many women need to increase their intake of folic acid, fortified foods, or dietary supplements, a recommendation of many health professionals. Some researchers suggest that a woman at high risk for neural tube defects should take 4,000 micrograms daily, but only in consultation with their physicians. Although folic acid supplementation is effective in preventing neural tube defects, there is ongoing debate relative to beneficial vascular effects associated with homocysteine lowering by folic acid supplementation. For example, Wang and others conducted a meta-analysis of 8 studies and found that folic acid supplementation significantly reduced the risk of a first stroke by 18%, particularly when supplemented over the course of 3 years, and reducing homocysteine levels by more than 20%. Conversely, Bazineau and others did a meta-analysis of 12 randomized controlled studies and reported that folic acid supplementation has not been shown to reduce risk of cardiovascular diseases or all-cause mortality among participants with prior history of vascular disease. Most recently, Clark and others, based on a meta-analysis of 37,485 individuals, reported that although folic acid supplementation reduces homocysteine, it has no effect on cardiovascular disease, cancer, or mortality in those with increased cardiovascular disease risk. Research suggests caution with excessive folic acid supplementation. A UL of 1,000 micrograms per day has been established for folic acid from fortified foods or supplements because megadoses could mask a vitamin B12 deficiency by preventing the developing of anemia that would otherwise be discovered by a blood test. Unfortunately, folic acid does not prevent nerve damage, so The B12 deficiency may lead to paralysis if not detected. Rothenberg indicated that this may be a problem associated with the mandatory folic acid fortification of grain products. Thus, individuals who may be prone to a vitamin B deficiency are advised to take a B12 supplement or consume B12 fortified foods. One advocate of vitamin supplementation to athletes has reported that runners need additional folic acid to replace RBCs that may be destroyed in heavy training programs. Unfortunately, no evidence is available to support this theory, nor are there any data showing that folic acid supplements will benefit physical performance. Only one study has been uncovered unrelated to folate supplementation to athletes. Matter and her colleagues provided folate therapy of 5 milligrams per day for 11 weeks to female marathon runners who were diagnosed as being folate deficient. Although the folate therapy restored serum folate levels to normal, no improvements were noted in VO2 max, maximal treadmill running time, peak lactate levels, or running speed at the lactate anaerobic threshold. To be sure, anemia resulting from a folate deficiency could have serious consequences for endurance performance, but a balanced diet, including fortified grain products, should prevent this condition from developing. All individuals should increase their intake of folate-rich foods, particularly vegetable sources, to obtain not only adequate folate, but other healthful nutrients as well. Women in their childbearing years should obtain 400 micrograms of DFE daily from a supplement to complement natural dietary sources, a recommendation approved by the American Academy of Pediatrics. Given the importance of adequate DFE, the National Academy of Sciences, in establishing the new RDA, indicated that women should get folic acid from fortified foods or supplements. Relative to the theoretical risk of vascular diseases, 400 microgram DFE again appears to be a reasonable amount that may be obtained through the diet, particularly with folic acid fortified cereals or with the use of a daily supplement. However, because several studies have linked daily intake of 1,000 micrograms of folic acid with increased risk of cancer, Liebman and Schart suggested that although women of childbearing age are recommended to obtain 400 micrograms of folic acid daily, males and postmenopausal women who take multivitamins might be advised to take one every other day and try to avoid cereals such as Total that are fortified with 100% of the daily value for folic acid. Folic acid has been combined with other B vitamins to evaluate health benefits of supplementation, which is covered in the section on the vitamin B complex. The adult AI for pantothenic acid is 5 milligrams, with lower amounts for younger age groups and greater amounts during pregnancy and lactation. The daily value is 10 milligrams and no UL has been established. Pantothenic acid is distributed widely in foods. It is found in all natural, animal, and plant products, but best sources include organ meats, eggs, legumes, yeasts, and whole grains. It should also be noted that highly refined processed foods have lost most of their pantothenic content. Pantothenic acid is an essential component of coenzyme A, which plays a central role in energy metabolism. You may recall that acetyl-CoA, which may be derived from carbohydrate, fat, and protein metabolism, is the principal substrate of the Krebs cycle. Pantothenic acid is also involved in gluconeogenesis, the synthesis and breakdown of fatty acids, the modification of proteins, and the synthesis of acetylcholine, a chemical released by the motor neuron to initiate muscle contraction. Except under experimentally induced conditions, deficiencies are not seen in humans. In such cases, deficiencies have been reported to cause a variety of symptoms, including fatigue, muscle cramping, and impairment of motor coordination. On a theoretical basis, pantothenic acid appears crucial to the active individual because it has an important function at the center of energy pathways. Several investigators have suggested that a deficiency would decrease the availability of acetyl-CoA for the Krebs cycle and thus shift energy production to anaerobic glycolysis, which is less efficient. Because efficiencies of or deficiencies of pantothenic acid have not been observed. Such effects upon physical performance have not been studied. Pantothenic acid supplementation apparently has no health benefit for a well-nourished individual. Liebman, in her review on how to select a multivitamin, indicates that you can ignore it on the label. Published research findings do not support an effect of pantothenic acid supplementation on coenzyme A levels or physical performance. Webster reported no significant effects of pantothenic acid supplementation of 1.8 grams per day for 7 days combined with allithiamine on 2,000-meter time trial following a 50-kilometer steady-state ride in highly trained cyclists. There were no significant metabolic, physiological, or psychological effects. Wall and others compared 1 week of supplementation with pantothenic acid 1.5 milligrams per day combined with icysteine 1.5 milligrams per day with placebo and found no effect on resting coenzyme A levels. Similar declines in post-exercise coenzyme A, lactate levels, and energy substrate use, and no difference in work output. Supplements of pantothenic acid appear to be relatively non-toxic. However, large doses of 10 to 20 grams have been known to cause diarrhea. The adult AI for biotin is 30 micrograms, with somewhat lower amounts for younger age groups and greater amounts during lactation. The DV or daily value is 300 micrograms and no upper limit is available. Good dietary sources of biotin include organ meats such as liver, egg yolk, legumes such as peas and beans, and dark green leafy vegetables. It is also synthesized in significant amounts in the intestines by bacteria. McMahon noted recent research suggesting that biotin may be involved in the regulation of gene expression, which may be related to its role as a coenzyme for a variety of enzymes involved in amino acid metabolism. Biotin is also involved in the synthesis of glucose and fatty acids. Because biotin is an important coenzyme for gluconeogenesis, it may have some implications relative to endurance performance. Deficiency states are rare, but may occur when the diet contains large amounts of raw egg whites. A protein in the raw egg white binds with biotin and prevents its absorption into the body. In such cases, symptoms include loss of appetite, mental depression, dermatitis, and muscle pain. For athletes who consume eggs for their protein content, it may be important to know that cooking the egg white eliminates the problem while providing the same amount of high-quality protein. It should also be mentioned in passing that raw eggs pose a risk of salmonella, a type of bacteria associated with food poisoning. Although a biotin deficiency could impair physical performance, no data are available to support this hypothesis. No research into the effects of biotin supplementation on health or physical performance has been uncovered. Thus, there is no evidence that biotin supplements improve health or increase physical performance capacity. Supplements of biotin appear to be harmless, as mock reports on no adverse effects in dosing ranges up to 200 milligrams daily. It would appear that biotin supplements are necessary for the physically active individual. Choline, an amine, is a water-soluble essential nutrient listed in the DRI report with the B vitamins, but it has not been classified as a vitamin. Commercial choline products are available as lecithin or choline salts, but the actual choline content may vary. Check the label for actual choline content. Choline is also marketed as a powder with carbohydrate and electrolytes to make a sports drink for athletes. An AI has recently been set at 550 and 425 milligrams per day for male and female adults, respectively. Lower amounts have been established for adults and children. No daily value has been established. The upper limit is 3.5 grams per day. Choline is found in most foods, particularly as lecithin in animal foods and free choline in plants. Milk, liver, and eggs are good animal sources, while good plant sources include vegetables, legumes, nuts, seeds, and wheat germ. Zeisel and Nikolouska note that normal diets deliver sufficient choline. Choline functions as a precursor for lecithin, a phospholipid in cell membranes. Choline is also involved in the formation of acetylcholine, an important neurotransmitter in the central nervous system. As choline is found in most foods as part of cell membranes, choline deficiency is very rare. The only research available involving individuals fed a choline deficient solution revealed the development of fatty livers and liver damage. Plasma choline levels have been reported to be significantly reduced following exhaustive exercise such as marathon running, and thus a possible reduction in acetylcholine levels in the nervous system may be theorized to be a contributing factor to the development of fatigue. However, exercise performance was not evaluated in these reports. Penry and Minor suggested that choline supplementation might only increase endurance performance and activities that reduce circulating choline levels below normal. As choline deficiency is very rare, the effect of choline supplementation on health status has received little research attention. However, its effect on exercise performance has been the focus of several studies in recent years. According to Zeisel and Nicolescu, several reports indicate that choline administration accelerated synthesis and release of acetylcholine by neurons. Research has shown that choline supplementation, either as choline salts or as lecithin preparations, will increase blood choline levels at rest and during prolonged exercise. Some preliminary field and laboratory research has suggested that increased plasma choline levels are associated with significantly decreased time to run 20 miles and improved mood states of cyclists 40 minutes after completion of a cycloergometer ride to exhaustion. In contrast, well-controlled laboratory research has revealed that choline supplementation, although increasing plasma choline levels, exerted no effect on either brief, high-intensity anaerobic cycling tests lasting about 2 minutes or more prolonged aerobic or exercise tasks lasting about 70 minutes. Given the evidence that choline deficiency states are very rare and that supplementation does not appear to enhance health or exercise performance, choline supplementation is not recommended. A balanced diet will provide adequate choline. There are 8 vitamins in the B complex, and because several of the vitamins in the B complex work together in energy metabolism, a number of studies have investigated the effect of either a deficiency or supplement of more than one vitamin upon health or physical performance. As noted previously, a deficiency of folate has been associated with elevated homocysteine levels. Deficiencies in vitamin B6 and B12 have also been associated with increased plasma homocysteine. The Consumers Union cited an analysis of 72 studies concluding that very high levels of homocysteine can provide heart attack and stroke. As noted previously, elevated levels of homocysteine may be a marker or result of disease, but not a cause. As might be experienced as expected from evidence presented on deficiencies on individual vitamins, a deficiency of several B vitamins together would negatively affect physical performance. This theory has been supported by studies in which daily intake was reduced to less than 50% of the RDA, such as well-controlled starvation experiments conducted during World War II. Vanderbeek and others compared a deficient group consuming about 1/3 of the Dutch DRA for thiamine, riboflavin, B6, and vitamin C with another group, consuming twice the RDA. After 8 weeks, aerobic power and onset of blood lactate accumulation declined by 9.8 and 19.6% respectively in the deficient group. In a later study, Vanderbeek and others compared 11 weeks of restricted, less than 55% of the Dutch RDA, and non-restricted levels of thiamine, riboflavin, and B6. Vitamin deficient subjects had decreased in VO2 max, VO2 at onset of blood lactate accumulation, and peak and mean anaerobic power, but there were no vitamin-specific deficiency effects on performance. The findings support earlier research showing a significant decrease in endurance capacity with a B complex deficiency. In 2004, the Consumers Union noted a growing body of empirical evidence involving the role of various vitamin B's or various B vitamins as a means to help prevent heart disease and other serious cardiovascular problems. At that time, some research had reported cardiovascular health benefits such as reduction in blood pressure associated with folic acid and vitamin B6 supplementation. However, subsequent research findings were not as promising. In the largest clinical trial, the Vitamin Intervention for Stroke Prevention study, a B vitamin supplementation of folic acid, 2 milligrams, vitamin B6, 400 micrograms, and vitamin B12 of 400 micrograms modestly lowered serum homocysteine levels but had no effect on recurrent stroke, coronary events, or death. Furthermore, in a study with heart attack survivors, those taking a combination of folic acid of 800 micrograms and vitamin B6, 40 milligrams, actually had an increased risk of heart attack and stroke. However, Schwamenthal and Tan noted that the VISP study had some limitations, and both studies involved individuals with cardiovascular disease. In general, research supports the idea that individuals who obtain adequate vitamins through a balanced diet will not improve performance through the use of B complex supplements. However, a number of earlier studies, several with children, have shown that when a deficiency state is corrected by vitamin supplements, physical performance is restored to normal. Moreover, some research with large doses of B1, B6, and B12, about 60 to 200 times the RDA, has shown increases in fine motor control and performance in pistol shooting. Bonk suggests that the beneficial effect was related to the role of these vitamins in promoting the development of neurotransmitters that induce relaxation. Additional research is needed to confirm this finding. Several of the principal Dutch investigators in this area, such as Bart and Saris, suggest that B-complex supplementation may be useful in sports with a high energy expenditure if these athletes consume large amounts of foods with empty calories, high sugar, and high fat foods. This again stresses the importance of eating foods that are high in nutrient value. Vitamin B6 complex supplementation does not appear to be needed for the individual consuming a balanced diet of wholesome, natural foods. However, consuming fortified foods or supplements to provide adequate amounts of all B vitamins for potential health benefits may be prudent. In particular, women of childbearing age should obtain adequate amounts of folic acid. Moreover, athletes should involved in intensive training for endurance type sports and consuming highly processed foods may benefit from a B complex supplement. Vitamin C, or ascorbic acid, is a water-soluble vitamin. Its alleged effects upon health and physical performance have been the subject of much controversy. The vitamin C RDA for adult males is 90 milligrams per day and 75 milligrams per day for adult females. Slightly lower amounts are recommended for children, whereas somewhat larger amounts are recommended during pregnancy and lactation. The daily value is only 60 milligrams, but the upper limit has been set at 2,000 milligrams per day for adults and lower amounts for children and adolescents. For current DRI, see the tables on the inside of the cover of the text. The best foods of vitamin C are fruits and vegetables, primarily the citrus fruits and leafy parts of green vegetables. Excellent sources include oranges, grapefruit, broccoli, and salad greens. Other good sources are green peppers, potatoes, strawberries, and tomatoes. One orange contains the RDA. Milk, meats, and grain products are low in vitamin C. Some products are fortified with vitamin C, but keep in mind when reading food labels that the DV is only 60 milligrams, while the RDA is 75 and 90 for adult females and males, respectively. A food level is indicating that one serving is 100% of the vitamin C RDA, would contain only 67% of the current RDA for males. Although vitamin C does not directly participate in enzyme catalyzed conversions of substrate to product, Jacobs suggests that it modifies mineral ions in the enzymes to make them active. Vitamin C has a number of different functions in the body, some of which have important implications for the physically active individual. Its principal role is in the synthesis of collagen, which is necessary for the formation and maintenance of the connective tissues of the body, such as cartilage, tendon, and bone. Vitamin C is also involved in the formation of certain hormones and neurotransmitters, such as epinephrine or adrenaline, which are secreted during stressful situations such as exercise. It helps absorb forms of iron from the intestinal tract, but about a 2 to 4-fold increased absorption, and is involved in the synthesis of red blood cells. Vitamin C helps regulate the metabolism of folic acid, cholesterol, and amino acids. It is also important in the healing of wounds through the development of scar tissue. Finally, vitamin C is a powerful antioxidant which helps it contribute to normal function of the immune system. Serious deficiencies of vitamin C are rare in industrialized societies because fresh or frozen fruits and vegetables are abundant. Also, the human body has a pool of vitamin C ranging from 1.5 to 3 grams. However, smoking, aspirin, oral contraceptives, and stress may increase the need for this vitamin, and a study by Johnston and others reported marginal vitamin C status in 12 to 16% of college students. The major deficiency disease is scurvy, a disintegration of the connective tissue in the gums, skin, tendons, and cartilage that may develop in a month on a vitamin C-free diet. Typical symptoms include bleeding gums, rupture of blood vessels in the skin, impaired wound healing, muscle cramps, and weakness. Anemia also may develop. It is obvious that many of the symptoms of a vitamin C deficiency we have cited would impair physical performance. Sensations of weakness could adversely affect all types of performance, whereas anemia would hamper aerobic endurance. Data available from several studies with vitamin C deficiency of subjects do suggest an effect, particularly the widely known Minnesota starvation experiments during World War II directed by Ansel Keys, as alluded in the previous slide. As far as supplementation for exercise performance, we'll talk about the antioxidant effects here in just a moment, but early research indicates improvement in individuals who are vitamin C deficient. There are some studies that indicate reduced severity of upper respiratory tract infections following ultra-endurance exercise, and there is current research showing no benefit to immune system functions after endurance exercise, and therefore no apparent effect on exercise performance. Vitamin C supplementation has been claimed to have significant health benefits, most notably the prevention of CHD and cancer. Because vitamin C is an antioxidant and because many related supplementation studies have included other antioxidant vitamins such as beta-carotene and vitamin E, we will discuss its effects on chronic disease and prevention of muscle tissue damage in physically active individuals later. A review by Weber and others suggests that adequate vitamin C intake may be associated with various health benefits for certain individuals. The amounts recommended approximate 200 milligrams per day, an amount easily obtained in the diet. Currently, the Consumer Union indicates that it is premature to recommend vitamin C supplements, but it does recommend a diet rich in fruits and vegetables, which, as previously noted, contain not only substantial amounts of vitamin C, but also other healthful substances as well. Eating the recommended 5 to 9 fruits and vegetables daily should supply about 200 to 350 milligrams of vitamin C. This is a solid recommendation for both the sedentary and physically active individual. The water-soluble vitamins consist of those in the B complex and vitamin C. Choline is also a water-soluble nutrient for which DRI have been established. In general, water-soluble vitamins are not toxic in excess. However, excess niacin may interfere with proper liver function, and excess vitamin B6 has been associated with neurological problems. Water-soluble vitamin deficiencies are rare in developed countries. However, supplements may be advised for some individuals. Women of childbearing age should consume 400 micrograms of folic acid daily to prevent the possibility of birth defects in the newborn. Elderly individuals should obtain adequate vitamin B12 through fortified foods or vitamin supplements. A water-soluble vitamin deficiency may impair physical performance, usually by interfering with some phase of the energy-producing process. In some cases, impairment may be seen in 2 to 4 weeks on a deficient diet. Supplementation with water-soluble vitamins has not been shown to enhance sports performance. And that concludes our discussion today. Thanks. I look forward to talking to you again. Have a great day. Yada, yada, yada.